Okay, and welcome back to the second part of the first lecture. This time we should be looking at action potentials and how we get the resting membrane potential, how the threshold is met and how an action potential is created. And we'll be looking at synapses and synaptic transmission. Okay, I want to take us back now to just some basic physiology of nerves. This picture here is a lovely, again, a micrograph of some neurons that have been stained for neurotransmitter. And in the small box on the right, you'll see all the little tiny green dots, and these represent individual synapses. So on one single nerve here, you can say hundreds and hundreds of green dots representing individual synapses. And it's believed that a nerve cell can have up to 10,000 synapses. Okay, and that means that the number of nerve synapses within the entire brain is something like 100 billion, apparently more than the number of stars in the galaxy, which I have no idea how they worked out, but apparently that's the case. Okay, so as well as neurons, we mustn't forget there are lots of other neuronal cells, nerve cells within nervous tissue. So this picture shows us a nice blue neuron, number 3, with the neuron cell body. The axon, number 2, going towards the synapse, which is number 7. Okay, but as well as this is an essential nervous system in the brain. We also have number 1 there, which is an oligodendrocyte. Okay, and that's the central nervous system equivalent of a Schwann cell. So that creates the myelination that speeds up the conductance of these axons. And it's very much implicated in MS, for example, where you get demyelinating conditions and plaque formations. So it seems to be a disease of the oligodendrocyte. Number 5 there in the yellow cell is a microglia, and these are sort of roundish. These are sort of the macrophages of the nervous system. Basically, they have a very much an immune function and release lots of cytokines and other sort of peptides, and migrate towards areas of infection or inflammation or damage. Okay, so they're the sort of macrophages of the nervous system. And then the green one here, we have an astrocyte. Okay, and these are very important cells because they surround every single synapse in the body, and they also contact the blood supply. All right, and these are integral in maintaining the perfect environment for synaptic transmission. Okay, and in fact, when we look at functional MRI scans, what we're actually looking for is the sugar uptake within astrocytes. Okay, so it's not actually looking at brain cell neuronal activity at all. It's looking at sugar and oxygen uptake within astrocytes, but it's a direct reflection of synaptic activity. Now, if we zoom into that sort of synapse, we see what we call a tripartite synapse. And this is made up of a presynaptic terminal and a postsynaptic terminal, the synaptic cleft, and then an astrocyte process, which is a sort of a process that comes around and surrounds the entire synapse and the cleft, closing off the environment from the outside. So this synapse surrounds the astrocyte process, so the axon synapse is surrounded by the process, as I said. And it very much aids removal of neurotransmitters and ions, specifically calcium, sodium, potassium ions, as well as neurotransmitters. So excess neurotransmitter will be vacuumed up by the astrocyte process, okay, and without which we'll get lots of dysfunctions within that synapse, because you won't have the ionic environment to discharge, and therefore it's implicated in lots of numerous sort of neurodegenerative disorders such as Alzheimer's, Parkinson's, etc. So the old-fashioned way of looking at nerves is literally a presynaptic neuron, synapse, postsynaptic neuron, whereas nowadays we're looking far more at the tripartite synapse and the role astrocytes and other nerve cells such as microglia are involved in making sure there's enough blood supply, enough nutrients, as well as the correct ionic environment for the synapse to fire. Okay, so you cannot have synapse without an astrocyte process. And here's a lovely picture depicting this. You can see the large sort of tree-like structure of this astrocyte surrounding millions of synapses and completely surrounding the nerve cell body there of a neuron. And in this electron micrograph, we have a similar picture. In blue, we can see an axon, presynaptic axon, with lots of vesicles of neurotransmitter, and in pink we see a postsynaptic dendrite and in between the dark lines show the synaptic cleft. And the green one here is the astrocytes we've just been talking about, completely surrounding that postsynaptic neuron and the neuronal cleft. Okay, right, so I just want to delve a little bit into some basic sort of nerve physiology here, and in particular action potentials and how they fire. So I'm just going to play you a short 2-minute video, which hopefully should explain it better than I can.
The fundamental task of a nerve cell is to receive, conduct, and transmit signals. Neurons propagate signals in the form of action potentials, which can travel great distances along an axon without weakening. To transmit an action potential over such a distance without weakening requires that the signal is continuously reamplified along the way. The central molecular players in this process are the voltage-gated sodium channels, which undergo a cycle of finely choreographed conformational changes. When an action potential passes, sodium channels open in response to membrane depolarization. Sodium ions rush into the axon, further depolarizing its membrane. Within a fraction of a thousandth of a second, however, the sodium channels switch to a new inactivated state, in which they are closed, but now also refractory to reopening. In this way, the membrane potential can recover quickly after an action potential has passed. The sodium channels then reconvert to the closed state, ready to be opened again when the next action potential is encountered. Let's examine the changing state of the sodium channel during an action potential. When no stimulus is present, the sodium channels remain closed, and the electrical potential measured across the membrane remains constant. However, if a depolarizing stimulus is applied by a brief pulse of electric current, the membrane will start to depolarize, away from the resting potential of about -80 millivolts. Some of the sodium channels will open, permitting sodium ions to enter the axon along their concentration gradient. If the depolarization is sufficient, even more sodium channels open, and the membrane potential rapidly approaches the equilibrium potential for sodium, about +40 millivolts. At this point, the sodium channels close, adopting the inactive conformation, where the channel is unable to open again even though the membrane potential is still depolarized. The sodium channels will remain in this inactivated state until a few milliseconds after the membrane potential returns to its initial negative value. The action potential is propagated along the length of the axon in only one direction. By examining the membrane potential and state of the sodium channels along a length of the axon, we can see why this is so. As a depolarizing stimulus reaches our section of the membrane, sodium channels open and current flows into the axon. This in turn depolarizes adjacent sections of the membrane, causing adjacent sodium channels to open, and the action potential is thus propagated along the axon. Sodium channel inactivation prevents the depolarization from spreading backward along the axon.
Okay, so we don't need to worry too much about the intricacies of that, but some of the main points I just want to reflect on are that we have a resting potential, okay, which is generally -70 millivolts, and this is maintained by slow passive leakage of potassium ions, as well as active transport mechanisms of the sodium and potassium. We need a stimulus to set it off, okay, and this could be sort of mechanical or chemical or thermal stimulus, okay, and if the stimulus is great enough and it's enough to set off the threshold, then you'll get a depolarization. Now the threshold is very important. In this case, we've got a threshold of -55 millivolts, because when we talk later on about low threshold mechanoreceptors or low threshold afferents versus high threshold afferents, we're very much talking about the sensitivity of this threshold. Okay, so a low threshold afferent will have a threshold closer to the -70, so more negative, such as -65, and therefore is much more easily set off. So a low threshold afferent takes a smaller stimulus to be set off and therefore depolarizes more quickly, whereas a high threshold afferent, so in this case it could be say -40, -45, it means it takes a bigger amount of depolarization from -70 to -45 to set off the depolarization. Okay, so the threshold level is extremely important, and if a stimulus is not big enough to reach that threshold, then you get a failed initiation. Okay, so when we come to high threshold afferents, these are nociceptors, meaning their threshold of firing is very high and they don't fire off very easily. Okay, whereas a low threshold afferent fires off extremely easily. Okay, and is very sensitive. Okay, so once we hit that threshold, we get an all or nothing response, causing a depolarization that will take it up to roughly +40 or beyond. Okay, and this is what we call the action potential. So depolarization only occurs if it reaches a threshold, and that threshold is only reached if the stimulus is big enough. So when we talk about all or nothing, we're really talking about the bit that's after the threshold. So a suprathreshold stimulus will be an all or nothing depolarization. Okay, whereas prior to that, if the stimulus isn't big enough, then it's then there won't be an action potential. Okay. Then obviously the sort of sodium channels close and the potassium channels open again, and we get repolarization, which drops below the -70 or becomes more polarized because the potassium channels are actually quite slow to open and close. And this part, the part of repolarization and the refractory period, is where the nerve cannot be set off again. Okay, and that's called the refractory period. Good. So that was action potential initiation. Now we want to look at synaptic signaling. So again, I'm just going to show you a brief video that describes this nicely.
Neurons transmit chemical signals across synapses, like the one shown in this electron micrograph. We can identify the dendrite of the receiving or postsynaptic cell, as well as two presynaptic nerve terminals loaded with synaptic vesicles. Note the narrow cleft separating the pre- and postsynaptic cells. The synapse converts the electrical signal of the action potential in the presynaptic cell into a chemical signal. When an action potential reaches a nerve terminal, it opens voltage-gated calcium channels in the plasma membrane, allowing calcium ions to flow into the terminal. The increased calcium in the nerve terminal stimulates synaptic vesicles to fuse with the plasma membrane, releasing their neurotransmitter cargo into the synaptic cleft. The released neurotransmitters diffuse across the synaptic cleft, where they bind to and open the transmitter-gated ion channels in the plasma membrane of the postsynaptic cell. The resulting ion flows depolarize the plasma membrane of the postsynaptic cell, thereby converting the neurotransmitter's chemical signal back into an electrical one that can be propagated as a new action potential. The neurotransmitter is quickly removed from the synaptic cleft, either by enzymes that destroy it, or by reuptake into the nerve terminals or neighboring cells.
Okay, so again, I just want to recap the important parts of that. So if we look on this diagram to the left, I've circled here the voltage-gated calcium channels. Okay, so the action potential will travel along the nerve until it hits that voltage-gated calcium channel, at which point it opens up and allows an influx of calcium ions. Okay, these are very important for drugs such as gabapentin, which you may have heard about, which is a strong neuropathic drug, and these seem to shut these voltage-gated sodium channels and prevent firing. So we get calcium influx that causes the vesicles to migrate towards the presynaptic membrane. Okay, the vesicles then fuse with the membrane and release their cargo of neurotransmitters into the synaptic cleft. Okay, and then these neurotransmitters then diffuse towards the postsynaptic receptors, which are ligand-gated ion channels. Okay, the ligand being the neurotransmitter, and these sit into receptors on the sort of protein gate and it causes a change in configuration of that protein, which opens the pore and allows sodium, potassium, positive ions to influx. Okay, this is a time-dependent mechanism, so no sooner has it started, the configuration changes, chucks out the neurotransmitter, and the pore closes. Okay, that neurotransmitter is then sort of vacuumed up by these astrocytes or by pores in the presynaptic terminal. Okay. So we've seen how an action potential will only occur if the threshold of firing is high enough. Okay, there are other mechanisms by which an action potential will occur or not occur, and one of these is spatial summation. Okay, so spatial summation is when you have numerous nerves or numerous presynaptic nerves all synapsing on the same postsynaptic neuron, and in this picture we have A and B are excitatory postsynaptic neurons in green, and the red one is an inhibitory we have the green ones are the excitatory neurons, and the red is the inhibitory neuron. Okay, and if the excitatory neurons fire, and here if we look at the graph at the bottom, we'll see how A causes a very mild depolarization, and that is what we call an excitatory postsynaptic potential, EPSP. Then if B fires, we get another small depolarization, which is another excitatory postsynaptic potential, neither of which reaches the threshold and therefore doesn't set off this all or nothing firing. When C fires, which is an inhibitory neuron, we get a hyperpolarization, so it becomes more polarized, so obviously it doesn't reach anywhere near that threshold, or in fact takes it further away from the threshold. So you can see how the culmination of the three, the ratios of the three, will summate if the excitatory potential, or the postsynaptic potential, is big enough to reach the -55 threshold. So when A and B fire together, or within milliseconds, the excitatory potential is doubled, and this is enough to hit the threshold and fire off an action potential. Okay, another type of summation is temporal summation, and this is the time lag between firing. So here we have a similar picture with two excitatory neurons and an inhibitory neuron. And if we look at the graph, we'll see how if A fires once, you get a small depolarization, but again not enough to reach threshold. Then if there's an adequate gap, then the resting potential is restored, and we get a firing of the excitatory neuron causes another brief depolarization, but again not big enough to hit threshold. When A fires in rapid succession, we get obviously a big influx of ions, and it's not quick enough for those ions to be removed via various methods, and therefore it has a cumulative effect, and this is often enough to summate beyond the threshold and cause an all or nothing action potential. Okay, so this last picture here, just showing the complexity of inhibitory neurons versus excitatory neurons, and how the firing of an action potential is dependent on the sum of postsynaptic potentials, the number of excitatory versus inhibitory ones. Okay, so obviously it's only if the excitatory outnumber the inhibitory and are big enough to reach that threshold will an action potential fire. So later on we come to look at inhibition within the spinal cord and sort of analgesia and stuff. We'll see that a lot of the excitatory and inhibitory neurons, the inhibitory neurons very much come into play. Whereas where we look at the way central sensitization occurs, there's a lot of increased excitatory firing. Okay, so that takes me to the end. Almost. Sorry, getting excited there. One last slide. This is just showing how not only is it the firing of the neurons that decides whether you get an action potential, but also the type of neurotransmitter. So at the top here we have a fast transmitter such as glutamate. Glutamate is the predominant neurotransmitter in sensory nervous system. It's used by all sensory nerves. Okay, far more ubiquitous than acetylcholine and noradrenaline. Remember, acetylcholine is what muscles use, and noradrenaline is what the sympathetic nervous system uses. So a fast transmitter such as glutamate will if fired at low frequency impulse, will give us these very brief but localized pulses, and if the neuron is activated with high frequency impulses, we still get brief localized pulses, but obviously in much more rapid succession. So a fast transmitter like glutamate can cause very brief, discrete impulses, and this is because the vesicles containing the neurotransmitter in the presynaptic neuron are housed very close to the voltage-gated calcium channels. So as soon as they're set off by the action potential, the vesicles can flood to the synaptic membrane and discharge their contents. This is very different to a slow transmitter such as neuropeptide, so substance P and CGRP. If you remember, we talked about peptidergic neurons, the C fibers. Half the C fibers are capable of releasing neuropeptides. Okay, and these neuropeptides, such as substance P, if the neuron is stimulated at a low frequency, these vesicles of neurotransmitter are quite far away from the voltage-gated calcium channels, and therefore the influx of calcium is not quick enough to set off the release of these vesicles if the frequency is low. Okay, when you increase the frequency, you get an increased buildup of calcium, and that finally reaches the vesicles of neurotransmitter, and therefore you get this kind of slow, diffuse buildup until they reach the synaptic membrane, and then once the action potential stops, you get a slow decay as well. Okay, so the difference between glutamate, which is a very fast transmitter, and neuropeptides, and this is very important as we come on to the sort of inflammation and sensitization. We'll see how neuropeptides are integral to this. Okay, so thank you very much. That's the end of the first lecture. Have a break, maybe have a sleep, and come back for lecture 2 later.